All right, well, it seems like everybody's already in here. So I'm going to start with our land acknowledgement. The Field Museum resides on the ancestral homelands of the Ojibwe, Odawa, and Potawatomi. The area was also a site of trade, gathering, and healing for more than a dozen other tribal nations. It is my absolute pleasure to welcome today's speaker, Dr. Frank Burbrink. Frank originally hails from Peoria, Illinois, so he is a true local. He reminded me last night that he's been coming to the Field Museum since before I was born. So he's been here and coming here a long, long, long time. Frank did his undergraduate and his master's work at the University of Illinois in Champaign-Urbana. And then he went on to do a PhD and a postdoc at Louisiana State University. Frank and I met first while I was down in Arkansas, but ultimately while I was doing a PhD in his lab in New York City at College of Staten Island, City University of New York. But for the last decade, Frank has actually been a curator at the American Museum of Natural History. He also was the chair of vertebrate zoology there up until July, and now he's done and he's real happy about that. Things about Frank are he loves snakes a lot. He loves doing field work a lot, and he loves writing papers more than anybody else I know. And he's authored well over 100 papers at this point. And a lot of his recent work uses whole genomes to answer questions related to evolution and adaptations in snakes especially. Frank is one of my best friends and collaborators, and he's the person who really taught me how to be a scientist and how to get a job, which obviously worked because here I am. I'm still, as my PhD advisor, he's somebody I'm still always trying to impress. So I ran out real quick, and so that I could match Frank, I got this FMNH tattoo on my hand. And you'll notice Frank has an AMNH tattoo on his hand. So Ariana got an INH one on her hand. Yeah. Ariana's over there. All right. And so I want to welcome Frank up to the stage, and let's give him a big warm round of applause.
All right. Thank you, Sarah. I really appreciate that introduction. Tell my wife, Lisa, she's good friends with my wife. Tell her, give that introduction to her so she can tell it to like the Parent Teacher Association and all this stuff I have to go to. Anyhow, this was the first natural history museum I have ever been to. I was here in the late 70s. And I remember coming back when King Tut came through the Chicago area, and then I came over to the Field Museum. So this is the place that I kind of got my start. But where did I get my start? I got my start in Peoria, Illinois, where that star is. Not the greatest place to get a handle on reptile and amphibian biology as a kid, but I've been catching snakes since I was 5 years old. And I would say it was all due to that field guide. That thing really changed my life. And it's what got me to even understand as a small kid in Peoria that animals have distributions that change over a landscape. So, I'd be sitting there in a cold, winter day in Peoria wondering why these snakes aren't out and why they are like at my relative's house in South Florida, having no idea how this works. And so that's what got me started. The other half of my life is music. So I play a lot of jazz guitar, but I'm also a mega record collector. And I just pulled these out of my 10,000 record collection of all weirdo stuff. So I am, those are the two main influences that sort of made me me. And then Sarah already had the introduction. I was going to say just a little bit more about me. I started at the University of Illinois in 1989, but I quickly moved to the Illinois Natural History Survey, where they let me work on Phil Smith's reptile and amphibian collection. What is really strange is Ariana Kuhn. Ariana, put your hand up. So she was a former graduate student of mine now, and now she's the curator down there in Illinois. So I feel like I'm seeding my people all over the US, and if we can get the Smithsonian and the CalCAD, we'll control almost everything. And then I moved, so I was that weirdo up there catching bats on permit, and then LSU. Went to LSU, that's in front of the museum there. And I was just telling Brian Crowther, one of my friends who's here and collaborators, that when I was at LSU, my advisor got fired. So I basically spent 4 or 5 years doing a PhD with no advisor. And so when my students complain about stuff and I'm giving free money for genomes and all these things, I'm like, you guys have no idea. I was like sitting there going through the Zoe record, by myself and trying to figure everything out. But it makes you kind of crazy and hard. And then I was at the City University for 13 years, New York, where I was Sarah's advisor and Ariana transitioned with me. to finally that weirdo that's at the American Museum of Natural History. So it's been a long journey and I'm back here in Illinois. So what I studied, generally all these things, when I was in college, I don't know if this still exists, but when I was in college, they used to say, you're either a taxon guy or you're a questions guy. You know, they only said it obviously in the male, way, but they would say that, and I was like, that's a really weird thing, like, this like false dichotomy. You either study a particular animal and you only do that, or you answer big questions, right? And I was like, that's stupid, just do them both, right? And so I think I can do all of these things that I say I do, but I probably wouldn't do them if there weren't snakes. So I'm really big time snake guy. And these are just some snakes that I've caught all around the world. So what I do and what I'm not gonna talk about mostly is I do a lot of expeditions to Brazil, sometimes a couple every year. And I work in this area called the dry diagonal. You see that kind of tan diagonal. 50% of Brazil is this dry savanna, seasonally dry forest stuff. It's not the Amazon rainforest or Atlantic rainforest. And this was part of a major NSF grant that I had with a bunch of institutions to figure out how animals adapt from the wet tropics to the dry tropics, right? So using lots of genomics and stuff like that. And this is just in the last 4 years. Those are all the expeditions that I've been on, like hitting everywhere. And this is like, it's not easy to do this stuff for vertebrates anymore and get them all back to New York City, but we do. Got awesome collaborators. This is what these habitats look like in Brazil, just really quickly. This is the Cerrado on the left and the Caatinga on the right. Lots of cactus, really crazy dry stuff. But what's really cool is like when you're out with Brazilians and you're on a collecting expedition, man, it is the most handsome people on the planet Earth. Look at that photo, man. Like, and I'm not denigrating my own country, but when I go out collecting with a lot of people here, it's like, you don't even want to look at these people, man. But these guys are like models, so it's spectacular. And we get lots of really cool reptiles and amphibians. Some are tetraploid. Some have these frogs with these giant Popeye arms. Really cool stuff. The only venomous frogs are in Brazil. I've caught a bunch of those. Those are the monkey tree frogs. Those are tetraploid. But it's, again, all about the snakes. And I was just on an expedition in the Chaco, which is in southern Brazil, adjacent to Paraguay. And the Pantanal, the biggest wetlands on the planet Earth, with your future curator, Diego Santana, who was actually a student of mine also. It's kind of like I'm not like the only guy that educates herpetologists, but for some reason here in the Field Museum, there's a lot of them. So this guy leads the best expedition. You hired full on the right person. This guy leads the best expeditions to the minute, which is a really hard thing to do if you're collecting in Brazil. He's awesome and he's really funny. Here he is yelling at a frog for some reason, and he's wearing an AMNH shirt. And I still have my feet in Madagascar, thanks to Sarah and Ariana, who did her PhD on all these Malagasy stuff. And then, of course, I still work in North America, and Sarah and I are out collecting and tissuing dead fox snakes on the side of the road and having a lot of fun. What I'm here to talk about today, okay, so things are gonna get serious. And so each time we get into parts that are a little bit more serious, I'll tell you like, it's a little more serious. It's gonna be, some rough road ahead, but I'll try to work it through. And if you don't understand it, we'll talk later. Or, you can go on with your life without understanding this stuff, I suppose. I think the most important thing that we found in phylogenetics in the last 10 years, and this is across all phylogenetics, is that the tree of life is not a bifurcating tree. It's a reticulating tree for lots of organisms. That is, you see hybridization between species or between groups, between non-sister species groups. And it's quite obvious and easy to detect this using genomic data. So in here, you see the red line connecting these two groups, right? That is not bifurcating, it's a horizontal transfer. And so you see this genomes being transferred between these organisms. And this is very common in lots of different things. And I'm only putting my papers up that have done this, but we found this, people have found this in bears, grapes, mosquitoes, blah, blah. This is Amavula ocellifer complex. These guys are kind of like whiptail lizards that you find in the US. These guys have reticulation everywhere. Probably one of the most shocking things is the pseudooxyrophiids, which are the gem snakes of Madagascar. We now know the whole genomes of all of these things, and it found reticulation through the entire phylogeny from 13 million years, you see those red lines, peppered throughout the entire phylogeny, right? We actually have all the genomes of these guys. Not even the lemur people that study those ugly ass lemurs have those genomes, right? And we've got them all. And so, and if you look at this, this is a reticulation through time plot, right? So you can see the number of, as the number of lineages increase through time, you get more opportunities for reticulation and you find more of this hybridization across groups. But what does this really mean in terms of speciation and what is happening? So there's a few endpoints in reticulation. And one is you're going to have the formation of a third species. That's possible. So you have the parental 1 and parental 2 hybridizing and producing a third thing. We see this in Madagascar. And sometimes that third thing goes on and diversifies and produces a whole bunch of other things. You can have collapse, which is extinction via hybridization. And this would be like what a lot of people who would be following biological species concept would expect, that if you have so much hybridization, it collapses and swamps the genomes and then you're back to one species, right? You could have full reproductive isolation, which would also be consistent with something like the biological species concept, where they stop hybridizing soon after speciation and they go their own way and they never hybridize ever again. And then what you see when you do, which is going to be about this talk, which is phylogeography. And I'll explain what phylogeography is in a moment, which is when you look at closely related species or newly formed lineages, they tend to have partial gene flow between them where they contact in space, right? And so this is partial reproductive isolation, which, if you get nothing else out of this talk, partial reproductive isolation is obviously an endpoint in speciation for things. That sounds weird, right? I think we think of speciation as being completely reproductively isolated, but for a lot of things, it's this. They continue to share genes between each other and out there among each other or between, depending on how many groups are involved. And I'll go over this, how all this works in a moment. These are the rat snakes. I'm just giving this as an example. My first paper on these guys was in 2000. I feel like I'm in some kind of weird time loop because every like 5 years I do something with these guys. I just can't get away from them. You can find them in Illinois. They come in lots of color patterns from the Chihuahuan Desert all the way to, you know, South Florida, all the way up to Vermont and New Hampshire, and then west over here to Illinois and Iowa. But they had all these stupid subspecies and all this stuff. And I did genetic stuff in 2000 with Joe Slowinski, who was a curator at the CalACAD. We found, and then most recently with genomic data, that definitely there are 4 species. The main split is at the Mississippi River, about 3 million years old. And so this is basically taking genetics and if you don't know what phylogeography is, applying genetics and then spatial tree inference to understand how these things have diverged over the landscape in a sort of tree-like structure. What I want to point out with this is that with all of these rat snakes, with all of these rat snakes, let's say you go down to the Mississippi, down in Louisiana, you see admixture, and those little pie charts are telling you how much of the genome comes from either the blue species or the red species, right? And so that's saying that individuals are the product of some sort of hybridization, right? Even after 3 million years, they don't let go, okay? So the question then should be, why don't these species collapse, okay? And to do this, we're gonna address 3 major topics here. One is the species ontology, concepts, methods. I know, like, that's not the funnest thing to sit and talk about, but we'll get through it. We'll pound through it, okay? The second thing is processes speciation. How are species really diverging, okay? And with the genomic data, it's telling us. And then this all comes down to genomic structure, and this is gonna be the last part of this talk. And I think this is kind of the big reveal that's putting things that we've known from PopGen up to like speciation phylogenetics level, okay? So it's marrying a lot of these concepts. So species are ontological individuals, and you can learn all of this from this paper that I wrote with Sarah and Brian. Brian's right there, who's also a co-author. This paper, for whatever reason, is on fire. People like to cite this because I think people may dislike subspecies as much as we do. So, but it goes through all of this stuff. And so first off, if you look at this, this is just a relationship tokegenetic net matrix, right, of interacting individuals that at one point separate into species 2 and species 3. And so what are ontological individuals? Well, species are individuals. They're like in the same way like us as individuals. They're not classes of things, okay? So an ontological individual, and this goes back to Gislin and Hull, who was here in Illinois also, of the 70s. And basically species are spatiotemporally bound. They exist on a landscape and they have a beginning and they have an ending, okay? And that beginning and that ending and how they relate to other species can be fuzzy, meaning they can still have interaction, right? And that is the properties of an individual. You as a 5-year-old don't quite look the same as you do now as whatever, a 40 or 50-year-old, right? But you are the same person through time. Think about that as species, how species demographies and all of those kind of things change through time. Those species are still the same thing, right? There are no instances of species. They're not classes, right? That is like, you know, hydrogen, if you have one proton and one neutron, you're hydrogen. You know, a particular species isn't defined exactly by the characters. They're defined through ostension, right? By pointing at them as being different. They're diagnosed. And they're cohesive. They're cohesive through this matrix, right? This tokegenetic matrix that is keeping them together. And all of the individuals in that species contribute to the mereological sum. That's a great word, mereological sum. That is the species, right? They're the individuals that make up the species that itself is an individual. Okay, last slide on this stuff. Species are lineages. They share a pattern of ancestry and descent. Here you have species A, B, and C with the colored lines representing their gene histories. Sometimes those gene histories follow that phylogeny, but other times because of gen processes that you can detect with incomplete lineage sorting relating to the population size, et cetera. Not going to get into that. The genes don't follow. And the genes also may not follow that because there's gene flow. Sometimes you have genes that move from C to B and make C look more closely like B, but that gene flow is okay. And so understanding species as lineages from the evolutionary species concept is consistent with species as individuals in terms of ontology. So these entities are discovered and they're genealogically exclusive. That's all I'm going to talk about on what I think a species is, and I hope you got something out of that. But there are a lot of Facebook biologists, I'm going to straight up say it, people that don't do anything else but get on Facebook and write about this is and this isn't a species. They clearly don't understand this, and I'm not trying to cause a war or fight anybody, but it's not that hard. Honestly, it's not that hard. Understanding what species are and aren't, there's always going to be this endless debate, but I think we've got it. And I know that sounds kind of arrogant. I don't mean it to be that way. But just check it out. It's not that hard. So let's go. So how are they maintained? So if you have gene flow, why don't they just collapse, right? Why don't they just swamp each other if they're still breeding, right? So shouldn't gene flow collapse these species? And so that's what this talk really is about, is how these species are maintained with gene flow. And the answer to it is gene flow is inconsistent across the genome. I think we've all been led to believe that when things hybridize and they back cross into the parental species, they just take that whole genome with them, right? But it's not true. Large parts of the genome cannot move between species, and I'll show you how that works. And instead of doing Brazil, we're going to work on the 17th most biodiverse nation on the planet Earth. 17th doesn't sound that exciting, but it is. There's a lot of awesome, great biomes in the US. And I'm going to walk us through some of the intersections of these biomes, because at the intersections of these biomes are where lots of species are diverging. And it doesn't matter if it's snakes or frogs or plants. These are constant barriers to gene flow that are generating species. So the first is, you know, the southeastern coastal plains connecting with the southeastern plains there at one. So you're going basically from the subtropics to the temperate regions of the US. Out here, as you move towards the temperate prairies, you get the temperate prairies, the semi-arid prairies, right? This is another major barrier. And this is a barrier where huge numbers of reptiles and amphibians turn over in that region. So that's another barrier. Another one here is going to be the intersection between the longleaf pines, so the end of the forest in the east, and the turnover in the semi-arid prairies. And then from the semi-arid prairies to the rocky Tamaulipan and mesquite habitat. Okay, and then finally, one of my favorites, which is the Chihuahuan Desert and the Sonoran Desert at the Cochise Filter Barrier. And if you're watching cowboy movies and they got those cactuses in Texas, you know they screwed up, okay? All right. And so just really quickly, we're going to walk through just a few of the taxa that exist at these barriers. And so the reason I'm using these snakes for this talk is to get an understanding of how they're diverging and maintaining their identities despite having gene flow. So we'll take the eastern rat snakes, not just the ones at the Mississippi, but we'll just do these guys here at this southeastern coastal plains. And using genomic data and self-organizing maps. So this is just an AI genomic clustering thing, actually made by one of my former students, Alex Byron at George Washington University. But it's basically like a structure kind of method, but it can use environment, it can use genomic data, it can use any data. And then you can see here, it tells you the probability of how many species there are, posterior probability, and what is defining those species. So this suggests there's 2 species, and it's mostly defined by alleles with a little bit of climate influence. Okay. Same thing in a new paper we just released on racers, same area, same thing, 2 species, mostly genomically defined. I just wanted to point out, this is Brian's guys at the Pituophis. You don't always get something. So I'm not saying if you just grab the genes from things that are spread out over a large area that you're guaranteed to get lineages. These guys don't do it. So I just wanted to point out that this isn't like a biased thing. Sometimes you find stuff that has no genetic break, right? The milk snakes, which was Sarah's dissertation, and we revisited with genomic data, have this big break in the at those semi-arid and temperate prairies. And then taking some other data from Chambers et al., we found, again, still consistent 2 groups. Moving down towards the longleaf pines, and we're only doing the corn snakes there in orange and blue. Those are the Slowinskii, Emerii, and Meow-morum. Again, same number of groups, or 4 groups. And then the Cochise Filter Barrier, Western Diamondback rattlesnakes are 2 things there. And those need a new name. That's coming up, but some people are going to probably go crazy about that, but whatever. And then the king snakes, right, which eat rattlesnakes, also break at the Cochise Filter Barrier between the 2 deserts. Okay, so now we know who this is and what they're doing. How does this all work? So we have speciation. So we're going to look at the timing of divergence, migration, and population size changes. This is basically the demographics of how things speciate. And I'll show you some images for that. And then maintenance and adaptation. Okay. So once you've detected 2 things and you know they're hybridizing and you know when through time and where spatially they're hybridizing, why aren't they collapsing? In that case, some genes should be correlating with environmental changes. Gene flow should only be occurring in well-defined hybrid zones. They're restricted to those. And there should be a subset of genes that are not integressing between the parentals. That is, they're not making it through the hybrid zone. Okay? That would show you that it's maintenance. Now then, I'm going to do that, and then we're going to find out why that's actually happening at the very end. So we'll take as an example, I'm just going to walk you through one of these species, and then we'll project that onto all of them. At least how we did it. And this is Pantherophis slowinskii and Emerii, meeting there between the blue and red dots. And that's a species I named after my good friend Joe Slowinski, who was a curator at the CalACAD. And this is a method that I developed that basically interpolates the whole genome admixture to find where the center of the hybrid zone is. And you can see that purple line helps me define where the center of that hybrid zone is. And then I'll get back to that in a second. So we take, to understand migration, when and how it's happening, we take genomic data, okay? And we feed them through what are called isolation migration models. And isolation migration models basically give you, using coalescent theory or diffusion modeling, gives you phylogeny, how things are related. It tells you when migration happens, so it knows what alleles are moving between species based on the coalescent, where they're coalescing. And it tells you the timing, if you know general genome substitution rate. And then the fatness, what most people aren't used to looking at is the fatness in these lineages, like how thick they are, tells you how populations are changing through time, okay? So here you have Slowinskii, and you see it diverges from the other 2. at about 813,000 years ago. So this is more than 200,000 generations, right? And you can see those black lines are telling you how much migration has been happening over time. And then you see that Slowinskii and Emeryii both have these population sizes increasing as you go towards time scale. That's basically how you read that. So that's saying, yes, there is migration between them, and that it has been fairly contemporaneous, at least into the late Pleistocene. We can look at these for all the species, and you can see all those different species that I showed you that have these connections. They all have wildly different shapes, right? But they're all generally more than 34,000 years old without collapsing. And they all show some migration. The thinness in these lines here is just a spatial visualization issue. They're still different from each other, and they're still showing little bits of migration. And most of these show some population expansion as you go towards the Holocene, towards time zero. Okay, so now that we know that these things are speciating a long time ago, that there's some migration and that there's some admixture, right? We know that you have individuals that are admixed. How are those boundaries being maintained? Because we know that migration's happened and hasn't collapsed these things, okay? So adaptive evolution has to be happening at specific loci, otherwise they would collapse. So genes should be significantly correlated, as I said, with the environmental changes. They should be at well-defined hybrid zones, these gene flow, and there should be something preventing them from integressing. So really quickly, you can use generalized dissimilarity modeling. It's basically asking, how does the genome change as the environment changes relative to space? So if genes just change relative to space, that's what you expect. As things get further away from each other, they should have more genetic differences. That doesn't mean they're species, right? But if they're changing very specifically to environments, that gives you an idea that the genome is being fit to these environments for an adaptive purpose. And for all of these, you can take geographic distance and hold it steady and understand what these other variables are doing. And you see in this case, all these bioclimate variables are changing one-to-one with the genome while factoring out space. So in this case, like with Slowinskii, the temperature in the driest quarter is a major driver of overall genomic change relative to space, relative to geographic distance, okay? Then we can do this other thing, which is called redundancy analyses. And basically, you're taking your genome, that ball there is your genome, all those little dots represent different genes. Okay, and you're asking, using a multivariate method, how those genes are correlated with those environmental variables significantly, right? And so this gives you all those colored dots tell you that those genes generally on the outskirts of that ball are related to those bioclimatic or elevational variables, or how much woody plants there are, et cetera. And you can take the identity of those genes and hold them back, right? So we'll take the identities. All these genes are massively selected and changing with these environmental variables, and we know who they are. So in your mind, just say we have a big pile of genes that are changing with environmental variables, and we hold those back, okay? Now then, let's add some space into this, okay? So as I said, I developed this thing that interpolates space over genomic changes. And I can find where the center of the hybrid zone is. And with the center of the hybrid zone, we can use these spatial Klein methods to say at the center of the hybrid zone, which is here 0, you have some spatial distance to a parental in this direction and spatial distance to a parental in this direction. And you can ask, how admixed is the genome over space? So if you're looking at that at the top, those are all parentals. And as you're going in the middle of the hybrid zone, you're sloping down to the other parental. Okay, and the inverse of the slope gives you the width of that hybrid zone, right? You can do that over the entire genome. And so for the Slowinskii and Emeryii, if you put both their ranges together, you're talking like 800 kilometers, right? With both their ranges. That hybrid zone is only 35 kilometers wide. It's actually quite small. So we take those genomes, and this is something another thing I developed called tornado plots. And this is basically gives you the idea of, if this is the Klein center, and that's the width of the Klein, and each one of those are dots, what are the genes that are concentrated and stuck inside that hybrid zone, right? The ones on the outside, these things may barely have diverged, or maybe a single snip in an individual. But in this middle of this tornado, right here, is telling you there's a whole bunch of genes that aren't moving outside that hybrid zone, okay? But that's interesting only if our hybrid zones look like the thing on the left. And they may not, because the United States and our biomes are pretty cracked up, right? If it was, you go from Slowinskii, a nice hybrid zone, to Emery, then that's fine. Those spatial methods will work nicely for you. But our hybrid zones might end up being like this, or they might have no parental species around them. They may be just disconnected. In fact, I think the milk snakes there are actually a disconnected and dead hybrid zone, not being fed by any parentals. So another way to get around this to say what genes aren't moving through those hybrid zones is to take space out of it, but actually understand space. That sounds weird. Let's just do this. Let's just rank the individuals by how admixed they are. So on the x-axis, that would be like all Slowinskii here. And then this is admixed all the way till you get to the other parental there, okay? All right, so that's your hybrid index. So we've taken space out of the x-axis. And then what we do is we basically regress each genotype through that admixture. So you're asking what genotypes change over as you go through the admixed individuals. Does that make sense? So you have this parental genome for like Slowinskii, and then you see these hybrid genomes are for a particular gene. And then it gets to the other species. So this is not space, this is how admixed. So you can see genes that go through admixed individuals, right? Genotypes that go through the admixed individuals, and they turn over really quickly when they get to the other parental. And so you can take this and graph and figure out what genes are changing over the genome rapidly when you go through admixed individuals. So these colored lines are all the ones that are really significantly changing. Like you have a genotype that is representative of Slowinskii, you hit the admixed individuals, and once you get on the other side of the admixed individuals, it rapidly turns over into the other species, okay? That gene is doing that. And the gray in the background is just the background genome that's not doing anything, okay? And this shows you there's a whole bunch of those genes. So take those genes and hold them back too, right? We got their identity. We've got the identity now with the RDA, the environmental guys. We could do simple genome scans quite easily, which is basically like using a PCA method with Mahalanova distances and asking what genes that define those two species are significantly different from the rest of the genome. And you can put a really high cutoff line, and all those genes above that red line are things that are massively significantly different, right? Again, without encountering space or environment. This is just saying they're heavily selected, okay? So now we've got all those methods that have identified a whole bunch of different genes, genes from the environment, genes that change over through space or through admixture, and genes that seem to be selected. And we can put them all together on Venn diagrams and find like, okay, there's a large percentage at least of the genome that I've sampled that are majorly significant, like here in 57, that are affected, that can be affected by all of these methods. The DAPSI is just another method that I put up there. I gave part of this talk at the evolution meetings, and when I showed this slide, some guy in the front row groaned. So, oh, I'm like, man, you don't have to look at it. You don't have to start growling at me from this slide. I'm just telling you, they all kind of do the same thing, right? So really quickly on this point, hold on, I'm getting hot, that the dates of origin of these things are between 161,000 to 813,000 years old. And you can have migration between 0.03 and 11 individuals per generation without collapsing. And we show that these loci respond massively to strong environmental changes, and that a subset of these loci sampled are maintaining these strong boundaries. Okay, so instead of, and I think we think of, because if we're doing taxonomy, we want a black and white world, right? We want to say this is this thing and this is this thing, but biology doesn't care about that. And you should care about biology over trying to force things into black or white, because the biology is telling you something much cooler is happening than trying to force things into stuff. And what a lot of people do is they want to say hybrid zones are areas where species are continuing to interact, and therefore they shouldn't be considered species, they should be collapsing. But I think the spin on it is hybrid zones are clearly filtration zones. That species here aren't collapsing after 34,000 generations. And you have disrupted gene flow at the intersections of these ecoregions with massive locus environmental selection. So you have a whole bunch of the genome that says it's not going through these things. And that's what's like maintaining the identity of these lineages that you can actually detect them. In fact, I would almost look at it, you know you have a hybrid zone and you know you have two things. Well, kind of done, right? You got your species. So that's the way I look at it. What I'm really interested in studying, this is the next major thing in my lab, using telomere to telomere genomes as adaptive integration. Why are the other genes getting through? There's some evidence that MHC genes are getting through, right? So immunological stuff is happening and that things are adapting to something on one side and those adaptations are being shared with the other species. And in fact, maybe the hybrid zone is why you actually see two things. Meaning if you didn't have a hybrid zone possible in this harsh environment with massive glacial cycles every 100,000 years, these things would be gone, right? But that they're borrowing adaptations from each other. This is a guess, but this is what we're working on now. Now, the last part of this is the structure of the genome. So I just wanted to point out, I've mapped all those genes to the genomes, and these are all the chromosomes and where these genes are in the chromosomes. And you'll notice something. There's a high proportion, a significant portion of these genes stuck on the Z chromosome. Okay, that's the sex chromosome. We need to know about the structure of the genome to know what's going on. Sex chromosome, something is really weird, always happening in sex chromosome. You have antagonism between male and female aspects, right? The antagonism means like, if you have genes that identify you as a male or a female on a particular chromosome, recombination will stop, right? Because you don't want to recombine the male and the female parts of those things, right? When you freeze recombination, what happens? You change your effective population size, rates of substitutions increase, you start putting genes that change how things identify each other as mates on those chromosomes. This list goes on and on. Haldane's rule, all these major, yeah, somebody likes Haldane's rule, man. They put their arms up, man. Hallelujah. All right. So all of this stuff. So then what is genomic architecture telling us? And so this is pretty wild. So if you go from the DNA and understand the three-dimensional structure of DNA and how genes are packaged in that DNA and where they live in the genome, as you're wrapping it up into 3 dimensions and then back into the cell, this is giving you the areas of the genome of where selection can really work against 2 species, where gene flow will not happen between those 2 species. And so let me point this out. So really quickly, recombination rules all, and I know this is, whatever, self-blowing my own horn, but this is a really good paper me and my collaborators wrote. I think it's really good. It's recombination-aware phylogenetics, which is the next step in phylogenetics and evolution, is understanding what recombination is like across the genome. And that's where you're going to get your species trees from, the trees that represent the evolutionary history without integration, et cetera. But just really quickly, back to your high school days or early college days with recombination. People don't like to think about this stuff, but because it's like, well, I don't do that anymore, but you got to. So it's like early in meiosis, right? You get recombination. You have a male and a female genome. You have crossing over, and that crossing over will put recombined chromosomes, right? So you have genes from one connecting to the other. If you don't have recombination and you have hybridization, those genes are never going to integrate into that genome. They're not going to move between those species. That's why I say recombination rules all. So remember, back to the rat snakes, back to these things here, these hybridization. Well, now I've done high quality whole genomes on everything. It's funny, in 2000, I started with pieces of cytochrome B, and now I have close to telomere to telomere sequence. It's a weird, personally, it's a weird thing to think about, right? If you told me this in 25 years, you would have whole genomes, I wouldn't have believed it, it's like, what did it cost Craig Venter to do the human genome? Like several $1 billion. And now it's like you get it in the next week. So without going through a lot of details, I built a model that
asked where are the genes that show allopatric selection, which is selection when they're isolated, or recurrent selection when they hybridize, and you get genes that get selected either to be borrowed or to be selected against. So sometimes they take those genes and they hybridize, and they're parts of those genes that change them, they tweak them, right? And so that's what this recurrent selection is, and they get an identity by tweaking them and making it their own gene after that. Anyhow, if you want afterward, I'll talk to how I developed this model. But if you look at, I've colored where most of those genes are, and there's like 400 gene regions. Again, the sex chromosome is the high point in this, and it might be hard to see, but you see this little black line running down the side of the chromosome? That's the recombination landscape. So not to get to the details, you can take a whole bunch of individuals, do pop gen on them, and figure out what the recombination landscape is like by how much genetic differences can happen, you know. Anyhow, I'll explain that afterward if you want, but you can get the recombination landscape by doing population level genomics, or you can get it by breeding a whole bunch of stuff and having multiple generations, much harder. And this is correlated, at least through all the snakes, the Lampeltinines, through 23 million years, this recombination landscape. So I just built this last week, just for you guys. Look at chromosome 14. That's showing you the recombination landscape, and you see at the end, it goes crazy, right? The recombination goes down to almost zero. You cannot have recombination in that chromosome. You got that? And then this is a thing that I was using called, it's basically a multivariate method to figure out where you have inversions in your genome. Do you guys know what inversions are? Like in one species, the genes are all going this way in a chunk, and in the other one, they're going the exact opposite way. If you have an inversion, you can't have recombination, right? Because you got the wrong genes going the wrong way when you have this recombination. And you see this area is a massive inverted area. FST is just simple population genetic measure of how different the genes are from each other. That goes through the roof. And then integration, you can't easily see that on there, but integration is dead in that area. So you have this like 3 megabase chunk of a genome just on chromosome 14 that has an inversion. There's no integration. I've mapped all the genes in there. There's 30 gigantic protein coding genes that do all kinds of stuff. They're dead. Those two things cannot move between those two species. Game over, right? You can look at chromosome 1, you kind of have the similar sort of thing. And then we go to sex chromosomes and it just goes through the floor, right? Recombination is dead through most of the things except for a pseudoautosomal region. That's not really an inversion. It's because you have a chromosome that's this big and one that's this big. There's no inversion. And FST is out of control. And if you go along that, it's hundreds of genes, right, that are not recombining between these organs. So when people want to talk about things not being species and stuff, there's no backing up from this. They're already species. The cool thing is that they're letting some of their genes through these genes. That's the next thing. And hopefully I'll see you guys at a herpetology meeting or evolution meeting, and I'll tell you what's making it through and why it's making it through. Really, the take home is reticulation is common. It occurs at all depths of the phylogeny. It's not a tree of life, it's a network of life. Speciation with gene flow is common in all organisms, and genome architecture and selection preserves these species boundaries, okay? Thank you very much, guys. And
I don't know if we have a microphone in here, but I think we're loud enough. We're gonna ask some questions. Taylor is there.
Hey, so, I think I was Pauline's rule. Do you have any examples of Pauline's rule in your hyperzones?
do I have Nah, because the problem is I don't know how fit anything is or sterile or not, right?
Well, I mean-
But the heterogametic sex are females and snakes, right?
Yeah.
So like, should I see, it would be great because where Brian and Mary live, they're dead center in that. So like, Brian, is the fecundity going down in female rat snakes where you live?
It's not my yard.
Not in your yard. Yeah, I don't know, right? Females should have a high probability of being sterile. There's another point to this and we should talk about it. It's too complex to explain. But those admixed individuals, none of them are heterozygotes. you have all these admixed genes, but they're homozygous for one parent or the other. So you go to this gene, it's homozygous for that parent. Go to this gene, it's homozygous for that parent. You go to the whole genome. That's your dead center of selection for those, because if you have heterozygotes at those genotypes, they're probably doomed, right? Because you don't see them. So I don't know about Haldane's rule. I would love to know.
No, I'm working on pairs for my dissertation, and I just found out 2 pairs of Haldane's rule where the species that don't currently naturally exist in the wild together, so in captivity, it's a 1 to 5 ratio, so there's only like
Oh.
1 female for every 5 males.
Interesting.
So I was wondering if there's anything like that in snakes.
It would be hard to know. Plus, we're also like in a landscape, again, that's really cracked up, right? Like really heavily developed with pockets of, habitat and stuff. And so I don't know how much of this is adaptation to like lots of human stuff as well in those hybrid zones. But my guess is given that when those admixtures happen, it's old. And it may be when the Mississippi River moves. I don't know if you guys know this, the Mississippi tends to move around like through the Atchafalaya Basin and then back to where it is. And stuff. So it ends up things that are on one side of the Mississippi end up instantaneously being on the other side interacting with something that they diverged from 3 million years ago. And so some of that may be overhang from that as well. So,
Thank you.
yeah.
Actually, what you just mentioned was what I was gonna ask you. The Wisconsin glaciation event pushed, the Illinois, we used to be the Mississippi River, it was too bad. And I was wondering if you see, an imprint of that, the genome of the snakes, how we look at it.
No, I mean, I think what they're responding to is, it's a bit older, but when the glaciers were melting over various times, Mississippi River was sometimes 200 kilometers wide, right? So it was like almost like an ocean isolating these things. But there are other areas, kind of what you're talking about, pushing them around. There's, Crowley's Ridge and the New Madrid Fault. You know the New Madrid Fault?
Yeah.
You guys know your screwed little thing, right? It's out here, you're close to it. So it's like the largest, I guess, one of the largest faults in the United States. But at one point, the Mississippi River shifted. And things that were on the east side were then all of a sudden on the west side. This was what, 1804 or whatever. That happened instantaneously. I would love to see what's going on in that genome play there as well. So yeah, you have a mix of the glaciers pushing stuff around, the Mississippi getting wider, and then all these other environmental things. I mean, at some point, you're not going to be able to tweak out every one of these things in the genome, because often major genomic events, like responding from the last glacier and expanding, wipes out the previous genomic events to some degree. You know a lot for a guy that studies human nutrition.
I spent 10 years in Peoria.
Oh, you did? Oh, God.
If you live there, you notice right away that there's a disproportionality between the bluffs and the size of the river. It doesn't make sense. It's because.
That's right.
The river shifted, Mississippi shifted.
You worked at Caterpillar or something?
No, I was at the University of Illinois College of Medicine.
Oh, okay. All right. Any other questions? You got it all? You got the basics? All right.
Thank you so much, Frank.
You're welcome.